This is my great pleasure and honor to visit this university, UAdeO, University Autónoma de Occidente. Particularly, especially my friend, long friend, Dr. Sylvia Paz, is now the top of this university. It's really a big surprise for me. I and Dr. worked together many years. So before I start my scientific talk, this is very beginning of our first collaboration, the product. On the top, left, you can see 1998. This paper published long ago. This is our first product of collaboration. Actually, I came here, Koreacan, to work with Dr. Sylvia Paz 2 years before. So first visit was 1996, I think. That is our beginning. And that time, we just started to investigate the special disease, natostomiasis, that's a parasitic disease, by eating fishes. Ceviche, maybe. Yeah. And that was a serious endemic disease. You can see in this map here, the many Mexican, particularly along the coastline, many people suffer from these parasitic diseases. And I brought the diagnostic method from Japan to Sylvia Paz, and we started to research. And the title of this paper, you can see many people in US, some of them sitting in the front. So I feel very happy to our reunion. And since that time, I and Dr. published many papers. This is a result of collaboration. It's a long history. So this is the basis. I have a chance to come on this stage. And today, not only this special parasitic disease, I talk about more general and common infectious diseases caused by parasite. So this is the beginning of my real scientific story here. The infectious diseases, old world and new world. Actually, I don't like this word. When you hear the old world and the new world, how do you feel? On this map, the old world, including the Africa, Europe, these continent, and the new world is considered Australia and Americas here. But this discrimination is a very narrow eye. So, oops. Okay. Actually, sometimes this separation works well. For example, in case of primates, monkeys, very clear differences between old world, oops. Yeah. The red one is the old world monkey area. And orange one is New World monkeys. So this application, New World, Old World, is very clear. But when we consider other diseases, we called Columbian exchange. Actually, conquistadors came from Europe to this area in South America and Central America, all Americas. And they brought many serious diseases to this side. For example, malaria, yellow fever, via Africa here. And also the smallpox. This is the most serious diseases to cause lots of damages in the American continent. Many people died, particularly in the central part. And on the other hand, these conquistadors bring back here syphilis, and that caused a problem in Europe, which spread to all the world. So those activities across the New World and Old World. This is another example, the slave trade from Africa to America around these ages. The slaves have schistosomiasis. That is an important parasitic disease, even right now, particularly in this side, American continent, the Brazilian people still suffering a lot. So these human activity and movement cause transport of so-called old and new world. However, when we say new, old, it's actually not true. This is how our continent, current style was established. Maybe you can see, I think most of even the students know the word Pangea. The whole continent started from just one plate that's been separated and established finally the current style. And when we consider this big one plate, many different creatures live on this area. Some are common, some are very unique, very narrow area. And the diseases also the same. Some are common, but some are very unique. If we look at this slide, this is not a disease. Just tell us wine, Venus. Even wine, it's called New World wine and Old World wine. In that case, the New World, Old World is quite different from the geographic separation. The wine started from around here, the Mediterranean area, then spread to Europe and part of North Africa. These are the old world wine producers. And China, which have a very long story, but they don't have a history of growing grapes. They don't produce the grape wine. They have their own liqueur, but maybe rice, some other grain. So in this aspect, New World, Old World separation is quite different. So I personally don't like to use old world, new world. And European people call the American continent people new world people, but it's not true. You have much longer history. This is typical example, no separation in new and old. This is the map of dengue fever. It's very common, transmitted by mosquitoes. I'm sure here, Koreakan, you also have dengue. It's kind of the tropical diseases. But in this case, all through the tropical areas, including Mexico here, Brazil, some Latin American countries, Africa, Asia, all through the tropical area, dengue is a problem. Which place is the origin of this disease? No one knows. We cannot tell. Even the genetic analysis of dengue in different places, they're exactly the same. And for dengue, different from COVID, we cannot develop the vaccine yet. So dengue is a serious problem. Maybe young generation sitting here, you can consider to develop dengue because it's your own problem and global problem. And still, we don't know how it moves. Just by limitation is a mosquito distribution. And global warming may spread this dengue more north and far south, expand endemic area. Few years ago, even in Tokyo, Japan, we had a dengue. Only one occasion, but in Tokyo metropolitan, we had a dengue. Fortunately, it's disappeared. But we don't know when it come back again. Yeah, COVID, of course, everyone knows that. And in case of COVID, it's very clear. It started from China, Wuhan, and spread all over the world in a very short time. Only 3 years, everywhere in the world, people suffer from the COVID. But fortunately, in this case, at the moment, people developed the vaccine for COVID. So at the moment, we can control this disease. But the problem of this virus disease is mutation. We don't know the precise mechanism, but already hundreds of mutants of this virus. So we are still afraid another serious type, virulent type, will appear and affect us. We don't know. So we still need a caution for that. So this is just a very simple but global the problem. And now, go to my professional area of parasitic diseases. I start with this liver fluke. I'm sure this parasite, it's a very tiny, maybe 2-3 millimeter length liver fluke, affects human liver, but not popular in the American continent. This is mostly the Asian problem. Particularly the Thailandia, where I used to work, is a very serious problem. And this, you can see, download from CDC homepage, the life cycle of this parasite. This liver fluke requires snail, freshwater snail, and freshwater fish. And when people eat the small freshwater fish in the river or lake, they get infected. And tiny fluke living in the river, we have already very effective drugs So as a parasitic diseases, we can easily control this one. But this parasite remain or repeated infections, no immunology, no immunological protection. So chronic infection, repeated infection many times, that cause finally the cancer. So liver fluke, small flatworm, infection through eating fish cause cancer. Actually, this parasite was recognized carcinogenic parasite. And this shows endemic area. Actually, this liver fluke, there are several different, maybe 3 different species. The one on the top, named Opistholchis viverrini, it's in Thailandia and Mekong River in this area. And another one is Chronorchis sinensis, mainly in China, Korea, and Japan also used to have this parasite. And in Siberia, another one, the Opistholchis ferineus. It's a mainly parasite of cat, but human also infect, and endemic in Siberia. So 3 different liver flukes in Asia, including Siberia, caused the cancer. So these 3 parasites is recognized as carcinogenic, making a cancer. But no information in the American continent. This is my friend in Ecuador, Manuel Calvapina. He found this parasite in his country. Very narrow, small village, but many people get infected. So it's very strange. You have this parasite as well. But why only in Ecuador, not other places in the American continent? Nobody knows. You may find in Mexico as well. I'm not surprised. Or in Brazil, not surprised. But no information at all. And maybe in the near future, this one also recognized as the carcinogenic liver fluke in this new world. The important information is molecular genetics. We have a tool to identify the relationship of all these infectious pathogens by molecular analysis to see their evolutional relation. And one surprise here is Opistholchis ferineus. This is the Russian one, Viverrini in Thailandia, and this is in the Asian ducks, the parasite. And Manuel Calvapina found Amphimels in Ecuador, was here. I think some of you can understand this kind of tree, phylogenetic tree analysis. If we consider the position of American one, it's rather older than the Asian one. It's unbelievable thing, because most of the pathogen, this kind of human pathogen or the mammalian pathogen, are considered originated from Africa, across the Asia, and jump over to America across the Bering Strait, and come down to South America. That is a general understanding of the movement of human and also domestic animal. Then why American one is older than Asian one? How can we explain? So although this parasite is not known in this area, how they go to Asia or Europe, it's a big mystery and very interesting part. How human move, or mammal, dogs and cats move. So we need more information, more materials to elucidate the relationship of these and its endemicity. This is another example. This is a lung fluke. Some of the Mexican people know this parasite. This parasite, the human infection occurs by eating crabs, shrimps in the fresh water. Now again, they need a snail, and then crabs, shrimps, and when people eat these infected material, they got the infection in the lung. So CT picture, you can see, looks like cancer, or sometimes pleural effusion, the pneumonitis. So nasty parasite. Okay? This is the crab, freshwater one. The small crab, if people eat, they get infected. And if the cat or dogs eat these, they get infected. Then recently, not only these, the crustaceans, but wild boar, wild pig, or deer, even deer, carry this parasite. So, still, maybe these paratenic hosts will be expanded. An interesting point is, in this, the lung fluke, we have varieties, different species by molecular analysis. So in America, particularly here, maybe I start here. North America, Paragonimus kerikoti. Here, Paragonimus mexicanus, Paragonimus inca. You have these parasites here. And different species in Africa, Africanus buterobilateralis. In Asia, Westermani, Scriabini, Miyazaki, heterotolemus. So genetically discriminable different species distributed all around the world. Again, molecular study here. It's very complicated, not straightforward phylogeny. On the top, Asian group here, African group here, another Asian group here, African group here, Americans here, Mexicanus, Colombia. These are American species. And Asian species down there. What happened? Evolution of these. Only these informations, maybe we cannot make a conclusion because it's so complicated. How they move, how they evolve in each area. Unfortunately, in the Americas, not many people work on this parasite. The Asian species, I and my friend David Blair in Australia worked on this and clarified relationship of Asian one, but it's not enough. We need more collaborators in Mexico or anywhere else, because it's quite common parasite in this area, maybe in the cat and the dogs. So you young generation of people can do this to make more clear movement of humans, animals, and the diseases. This is another one. I think this is quite common in dogs and cats. Even here, we call it dipididiasis is the name of disease. The parasite is Dipidium caninum. Caninum means dog parasite, canine parasite. It's not so big parasite and not serious disease. But see, you can see here, cats, dog, and this parasite transmitted by fleas of dogs or cats. So naturally, the parasite only keep life cycle, fleas and dogs and cat animals. But if people ingest this flea from dogs and cats, people get infection. Usually people don't eat these small fleas, but the babies, niños, accidentally get infected. So most of the human infection is a baby, crawling very small children. So pediatric textbook, we can see this disease. More than 95% of human infection is a baby. And this is distribution. Now go back to the world. You can see all over the world, particularly in Europe, it's very heavy, common in dogs and cats. All over the world, where dogs and cats is there, we can find this parasite. So it's a global parasite. But if look at the human case, accidental, the baby infections, it's very limited. Very strangely, Europe and North America, United States, they have many cases, maybe over 100 cases. But in Asia, dogs and cats carry this parasite, but human, very, very low. Even in Japan, we have only 17 cases. And Mexico reported cases, only 5. Recently, I found this interesting case report from Mexico. The doctors are all Mexico DF, Mexico City. But this only one case report appeared. See, top right, very highly ranked journal, British Medical Journal, the United Kingdom, very highly ranked medical journal. This case, only one case reported. So these doctors were very lucky. But what I recognized is, in this paper, patient, 18 months old, female infant from Sinaloa, Mexico. So the doctors, medical doctors, pediatricians in Sinaloa missed good chance. If they pay more attention, I'm sure you can find many cases of children infected. And if you investigate the infection in cats and dogs, you will find this parasite quite easily. But no information in this area at the moment. So this is the one thing you can consider. This is I and Dr. Sylvia Paz's very old works. And I'm sure still it's a problem. Natostoma is a disease of the skin, sometimes like this, sometimes moving swelling on the skin. And infection is eating mostly fish or occasionally shrimp. And the natural cycle without human, but maybe dogs, cat, and pigs. So if human, we eat freshwater fish, ceviche, or some other dishes, you may have a chance to get infection. So this is a fish-borne. So where people eat fish, you may have a chance to get infection. And the parasite itself is in the stomach of dogs and cats or the pigs. This is the endemic areas on 2009. So mostly in Asian country and a few African country, and here, Mexico, and here. But now this parasite expanding, that means more people eat fish. Maybe the nutritional reasons. Fish are considered to be healthier than meat. So fish eaters are increasing. And you can see these red areas, Mexico, Japan, Thailand, and Vietnam, are very high incidence because local people also still depend their nutrition on the fishes. I just want to say here, Mexico is now still leading top of this disease in the world. And sometimes this parasite go to your eyes and cause serious diseases. But strange phenomenon is here. Japan, Thailand, India, Mexico, we have many cases affected eyes. But only in Thailand, they have problems of brain migration. The same parasite. Why only in Thailand they go to the brain? No one knows. Some ethnic relations, or parasite in Thailand is different? We don't know. And very recently, this is part of my work, we found many cases in here, Myanmar, next of Thailand. They have many ocular affections, but fortunately no brain affection. So again, yes, some strange things there. Thailand, Myanmar, just across the border, but the type of disease is different. And maybe the one reason is this table. Natostoma have many different species. See, from this dark line above is the American species, and below is the Asian species. So you have 7 American type natostoma and 6 in Asian type. And only among these 7 different natostoma species in Americas, only one, Natostoma binucleatum, is a pathogen for human. In Asia, 4 species are identified as human pathogen. And this is I found in Sinaloa with Dr. Sylvia. The very unique species. We found this Natostoma truchidum, very big, over 10 centimeter. It's quite unusual as a natostoma. Most of natostoma is a very chiquito, small one. And this parasite from opossums in this state. Again, molecular studies, PCR sequencing revealed this big one, Natostoma truchidum, far away from other species, Natostoma species. From this tree analysis, Natostoma truchidum supposed to be the ancestral, very original species. Is it true? How, if this is true, this species moved to all other countries in Asia and then differentiate. How is it happen? So molecular analysis, PCR sequence, is a very powerful tool, but often it brings us new questions and very difficult to answer for this. In this case, we only examined ITS2, maybe COX1, also the same. But some other genes which we don't know might cause the different differentiations. So maybe the whole genome analysis of these, including other American species, you have 7 different species, may give some answer what happened. So molecular tool is useful for diagnosis, but gives new questions, one after another, about the phylogeny or epidemiology. So many things we can do it, particularly due to COVID pandemic. Everywhere in the world, PCR sequencing machine becomes available. So maybe it's not so difficult to do more works on this kind of unique distribution of parasites. Maybe this is the last one. I'm not sure whether any one of you have this very interesting parasite, Diactophyma renale, commonly known kidney parasite, affect the kidney of dogs. Like Natostoma truchidum, this nematode, very, very big. Also, it's a dog parasite. In kidney of dogs, it's a very tiny organ. But the female adult one is over 1 meter. Amazing size as a parasite. And life cycle, only very simple. Maybe fish, reptiles, the intermediate. And if animals, dogs, eat these, they get infected. And very serious problem of the kidney disease. This is actually parasite get from the kidney of dogs. See, this is dog. One dog has this many gigantic parasites. And this parasite, because it's so big, very well known in ancient times. This book was published 16. I think it's the time of the Renaissance. Already very clearly described by etching picture. So very old known parasite, biggest parasite, nematode, and still more ancient evidence. This is kind of fossils, coprolite, feces of maybe dogs, becomes a stone, and which contained the egg of that parasite. And it was before Christ, 3,000 years. So it's very strange parasite, long history, but not big distribution. And all over the world, human accidental infection, it's only 18 cases, all over the world, not many, just accidental cases. This is the distribution of human cases. America, 1 case in Brazil, 2 cases in Peru, many European, the Mediterranean, the Asians. No special hotspot, very widely distributed. But this is the literature survey I and my friend done. If we look at cases of dogs or other wild animals, this place, very, very high, particularly in the northern part of Brazil, many, many cases, not many in Asia. In some part of China we have. So it's very different distribution, human cases and animal cases. Usually humans and dogs live together. But this distribution tells big difference in distribution. Why not many human cases in Brazil? They have hundreds of cases in dogs, but only 1 human. While the Americans, not high in dogs, but they have more cases in humans. So this is another big mystery. And I checked the literature from Mexico. No report, not much report in dogs and humans, very low. Very close to these areas, but very different distribution. We really don't know. So many questions here. How and where that this parasite is maintained? And in the textbook, it said fish-borne parasite. When people eat fish, get infected. But the geographical map tells no relation to fish diseases. If it is really a fish-borne diseases, Mexico should have, because we eat ceviche. See? So it's not definitely not fish-borne diseases. Maybe some other route of infection to humans. And no genetic information at all, whether European, American, Asian, really genetically same species or not, nobody knows. So this is the end of my talk. I said, although the cultural and the human movement, we have the borders, but diseases, particularly these infectious diseases, are borderless. They easily across country, easily across continent, spread all over the world. Maybe different speed and different requirement of the vectors. So, as I showed some example of parasitic diseases, we need collaboration across the border. Just I and Dr. Sylvia Paz did nearly 30 years, and we still left many questions. So young generation should work hard to solve all these questions. And I hope under Dr. Sylvia Paz, people in this university work this kind of topic in the future. I sincerely hope that. Thank you so much for your attention.
Con ese aplauso le agradecemos al doctor Yukifumi Nawa esta gran disertación. Vamos a pasar ahora a una ronda de preguntas o comentarios que quisieran hacerle a nuestro conferencista invitado. Es cuestión solamente de alzar la mano. No miraba al doctor Juventino. ¿Nos pudieran apoyar, compañeros, con un micrófono, por favor? Muchísimas gracias. Adelante, doctor.
I just have one question.
Yes.
Are there any hypotheses trying to explain why some of the parasites found in the so-called New World can be like genetically older than the other parasites found everywhere else?
By now, no answer for that. And the problem is that at the moment, most of the molecular phylogenetic analysis based on the very tiny sequence of mitochondria and usually ribosomal DNAs. It's a very tiny part. And we don't know the whole genome of most of these pathogens. So these tiny markers are currently used, ITS2 and COX1 and NDH, maybe not really reflect the phylogeny. Maybe whole genome analysis is required. Because these tiny markers, very useful as a marker, quite because the mitochondria and the ribosome shared everywhere, and very easy, stable to do the PCR. And we have already big data bank. But those are only very tiny part. And DNA, the mitochondrial DNA and the ribosomal DNA is a very essential gene and maybe not change much. So rather stable. So we only based on the stable gene as a marker to draw the phylogenetic trees. But if we look at another genes, such as the genes related to the morphology or the body structure making markers, we may get a totally different result from this. So we need more and more data for that. Maybe the whole genome analysis of everything will give some advanced answer for that. Until now, no idea how the creature moved to the old world, so-called old world. No answer, I think.
Thank you.
Si alguno de ustedes quisiera hacer alguna pregunta en español, con todo gusto le traduzco para el doctor, con toda confianza.
Por allá levantaron la mano, doctor.
Adelante.
First of all, thank you for your amazing talk. It was very interesting and it was a lot of information to process. And my question is, if there is a possibility that the new appearance of these parasites could be associated with genetic resistance that humans previously have, and due to the new communication between old world and new world, could be this genetic resistance disappear or go inactivated by epigenetics?
That's a good question, but very difficult to answer, particularly the genetic resistance to parasitic diseases. The very famous one is susceptibility or the resistance to malaria. If you have some blood type, the mutations, Mediterranean thalassemia is a resistant. Typical example, many of the African people have thalassemia. And also some Asian countries like Thailand, they have resistant genes, G6PD mutation. They also have the resistance. So those protozoan parasites, we have some cases we have good example of resistance, but generally still we don't have enough information about the genetic difference in the resistance, particularly to those hermits, big parasite like Ascaris or Natostoma or whatever. These big parasites, our defense system, immune system, also very complicated to fight against these big ones. Protozoa parasite is a tiny, microscopic level, smaller than the cells. Malaria is infected into the red blood cell. It's a very small. In such case, maybe resistance developed rather easier. But helminths, very difficult to analyze the resistance susceptibility. So while I have been working in Thailand, I asked the parasitologist why only in Thai they get infection in the brain with natostoma, but they don't have any answer. And so far, the molecular analysis of natostoma in Thai and natostoma in Mexico, binucleatum, not a big difference. So from the parasite side, difficult to say the difference. Maybe human, we really cannot find yet the basis of the different susceptibility. So we have to ask natostoma parasite, why you go to brain, why you go to skin? Actually, the migration of parasite is an interesting question.
Thank you so much.
¿Alguien más? ¿Alguien más que quiera hacer alguna pregunta? A ver, acá, ya vi. Ahí va.
Hi, I just want to say something. I want to thank you because we really appreciate that you have the time to talk about this in our university, and it's really interesting for us because we have some classes for that. And actually, my friend and I were talking about how the information that you gave us today, we actually learn from internet about it, and thank you so much. That's it.
Tenemos espacio para otra pregunta.
A ver, de este lado nadie. ¿Por acá? Parece que no. No sé, ¿alguien más? Adelante.
Solamente deseo hacerles un comentario a ustedes. Creo que la conferencia del doctor Yukifumi Nawa nos plantea un panorama muy claro acerca de lo que ustedes, principalmente los jóvenes, deben de hacer en esta etapa tan importante de su formación profesional. Es decir, no nos debemos de quedar solamente con los conocimientos o lo que sucede en Sinaloa o en nuestro país. Él claramente mostró cómo hay algunas enfermedades que empezó hablando del dengue, cómo hay algunas enfermedades que son compartidas en todo el mundo. Hay diferencias en otro tipo de enfermedades y por supuesto los agentes causales de las mismas. Y si bien esta plática se enfocó más a parásitos, también se hablaron de algunos virus, como el propio virus que ocasiona el dengue y COVID-19. Esto también es válido para otros microorganismos como las bacterias. Entonces, prácticamente esta plática es una invitación argumentada de que los conocimientos tienen que ser globales. Siempre tenemos que pensar en todos. Y pues eso es lo que a mí me gustaría que se les quedara también de esta plática, que en resumen, que hiciera crecer más sus alas, que es lo que más me interesa a mí. Muchas gracias.
Con este comentario muy afortunado que ha hecho la doctora Sylvia Paz Díaz Camacho, vamos a pasar al siguiente punto. Y como bien dice ella, estas gestiones son a favor del conocimiento que adquieren aquí, en esta universidad, todos ustedes linces. Vamos a hacer entrega, antes de pasar, la persona que nos estaba ayudando con pasar el micrófono, y si ustedes ocuparan asistencia, es el director del Centro de Lenguas Extranjeras de nuestra universidad, del CELE, el doctor Edgar Adán Valenzuela García. Muchas gracias, doctor. Muchísimas gracias por la conferencia que nos ha brindado a la comunidad lince el doctor Yukifumi Nawa, y para hacer tangible este agradecimiento, la Universidad Autónoma de Occidente le extiende un reconocimiento. Para ello invitamos a que nuestra rectora, la doctora Sylvia Paz Díaz Camacho, líder de esta casa de estudios, haga entrega del mismo. Mientras ella sube acá al escenario, me voy a permitir ir leyendo este reconocimiento. La Universidad Autónoma de Occidente otorga el presente reconocimiento al doctor Yukifumi Nawa por la excelente disertación de la conferencia magistral Enfermedades Infecciosas del Viejo Mundo y del Nuevo Mundo impartida en el Teatro Lince de la Universidad Autónoma de Occidente. Agradecemos su valioso aporte y amable disposición para compartir su experiencia y conocimiento. Culiacán, Sinaloa, 12 de diciembre de 2023, por la cultura, la libertad, firma la doctora Sylvia Paz Díaz Camacho, rectora de la Universidad Autónoma de Occidente. También le extiende un detalle de parte de la comunidad Lince. Y pues este momento no puede quedar sin una fotografía que quede para siempre de este momento. Vamos a pedirles a todos ustedes, por favor, se pongan de pie y se unan más hacia el centro. Mientras pedimos a quienes presidieron este evento y autoridades, por favor, suban al estrado a acompañar a nuestra rectora, la doctora Sylvia Paz Díaz Camacho, y al doctor Yukifumi Nawa, nuestro conferencista invitado, para que puedan hacer esta toma de este evento. Sí, ahorita la una, ¿verdad? Okay. En estos momentos se están acomodando los linces, se están acomodando acá nuestras autoridades, están nuestros camarógrafos, nuestras demás autoridades. Y en eso, 1, 2, 3. ¿Todavía se están acomodando, compañera, otros jovencitos? Listo. Listo, linces. 1, 2, 3. Eso, linces.